coming to this Dean's Lecture. And I think probably everybody in the room knows that the Dean's Lectures are to celebrate the appointment or promotion of someone to the rank of professor here. And you know, I say it every time, but it's true that it's very, very difficult to become a professor at Johns Hopkins. And we're one of the elite universities in the world, and it's hard to get appointed to the faculty, but it's even harder to become a professor, because to do so you have to get the affirmation of your peers from around the world. And you know, it's not our decision in a sense. It's the decision of our colleagues in academia and sometimes in NIH and foundations around the world. So it's a big deal, and so we're here to celebrate that today. And you all know Thomas Hartung, who directs the Center for Alternative Animal Testing, and is the inaugural Durham Camp is a Bindon Chair for Evidence-Based Toxicology in the Department of Environmental Health Sciences. And when he first came, and then we received that gift to elevate him to that, we had a similar function then, which was enjoyable, and I'm looking forward to today. Let me just tell you a little bit about Thomas that you may or may not know. But he is originally from Germany. He joined the faculty at the University of Constance in 1994 as an assistant professor of biochemical pharmacology, and was there until 1999, and then became an associate professor. He's been an honorary full professor of pharmacology at Constance since 2003. From 1996 to 2002, Thomas served as the CEO of the Steinbeis Technology Transfer Center for in vitro pharmacology and toxicology. And in 2002, he became head of the European Center for Alternative Methods, ECFAM, at the European Commission Joint Research Center in Italy. As head of ECFAM, he was a leader in Europe and around the world. He was integral in accelerating the alternative methods validation process and in establishing a network of more than 400 experts from all stakeholder groups to facilitate global regulatory harmonization and toxicity testing. And since he's left, he's been a well-regarded and outspoken commentator on those processes. He received his PhD in biochemical pharmacology from the University of Constance, and his MD with a dissertation in toxicology from the University of Tübingen. He completed his medical internship at the University of Freiburg in surgery and at the Hospital of Singen, Germany. And for everybody who knows him, you know, he's a man of incredible talents. I mean, he's a magician, stand-up comedian, a photographer, all of which stands you in good stead as a Hopkins faculty member. So Thomas, we're looking forward to hearing the sneak preview of the beta release. Okay.
Okay, as long as the speaker doesn't disappear. Okay, it's wonderful to be here. It's wonderful that we can celebrate jointly my appointment to the faculty, and I'm really grateful for having this opportunity to work with you. Toxicology, especially regulatory toxicology, is somewhat outside of the academic discussions usually, though it is obviously a very applied science and very important science for our society. Arnold Lehmann was head of the FDA in the 50s, and he's well known still for a quote which is, "You too can be a toxicologist in 2 easy lessons, each of 10 years." So I took quite exactly these 20 years, these 2 lessons of toxicology. It's about 20 years since my MD in toxicology. And as a brave scholar, I did learn the first 10 years how to do toxicology. And then I learned the second 10 years what is not working so well in toxicology. And I hope to spend the next 10 years here to help improve it. So for the students here, this can be, if you're not in the division of toxicology, the very first day of 19 years, 11 months and 30 days of becoming a toxicologist. For those in the division of toxicology, it's less, and you know the years here in our division count twice, yeah? So it's getting you much faster there. Okay, I have to speak a little bit about the regulatory toxicology version 1.0 and 1.1, 1.2, which we have seen evolving over the last, let's say, 100 years. Because you need to understand why we think that a version 2.0 is necessary and has to be brought about. So what are we talking about? We are talking about consumer products. We are talking about various industries, and what you can see here is chemistry, you can see medicines, cosmetics, food. These are major industries and major exposures everybody is exposed to. And it's major business. Worldwide trade is about 3 trillion each for chemicals, for medicines, and for food, and about 200 billion still for cosmetics. And obviously, we want these products to be safe. Safe not only for us, but also for the more vulnerable parts of the society, as shown by this baby. So what have people been doing over the last 100 years? While introducing increasingly substances into these products, they have started to test, to avoid problems. And every scandal alerted us of something else. And we were using animal tests mainly because this was the life science technology at hand at the time when we devised this toxicology. And we are talking about an industry itself testing on animals, which we use as the approach for safety assessments of substances, is a $3 billion enterprise per year. So roughly 0.1% of the turnover of the regulated industries is spent on quality and safety, on safety assessments of their components. So if you look, environmental health sciences is all about environmental risks, obviously. But it's not only about the risks, it is also about the perception of these risks. And this is the reason why we demand nowadays a lot of testing. People are concerned about environmental chemicals. This is a survey from Europe in November, but there's similar data, but much older, from the US available. And what you can see here, if you can read from the distance, is that environmental pollution and damaging your health is very likely, feel many people, 18% of the people feel, and 43% say it is fairly likely that their health is being damaged by environmental pollution. In comparison, getting a serious illness is much less of a concern. Though everybody will have a serious illness, which is called death, at some point in their lifetime. So people are more concerned about environmental pollution damaging their health than they are about serious illnesses. So this shows the tremendous attention we give to this field. So we're using animals for the substance safety. And there's a balance to be found between using animals and having them suffer, but it is also a balance to be found about the quality of predictions, the throughput, and the costs. And I will touch a little bit on this because these are all three driving forces at the moment to go away from animal testing. And people less and less accept that we are using animals. These are Gallup polls from 2001 to 2008. And the question posed to the US citizens here was, is medical testing on animals acceptable morally? And while in 2001 it was 65% who said it's morally acceptable, this fell until 2008 to 56%, while the number objecting did rise from 26 to 38%. So there is also societal pressure to use less animals, which is the basis of our foundation of toxicology. And these protests can be quite vigorous. And as you can see here, quite nicely coined, thanks to animal research, they'll be able to protest 23.5 years longer. You might argue whether the values are right, but society is in a certain uproar against these approaches. So we have a demand of society to produce safe products, higher and higher safety standards. It's getting less and less acceptable that 1 in a million patients treated dies from the treatment, which has been our previous threshold. But at the same time, they want to do these things differently. And there's good reason to be skeptical about products. This is from the early times of drug industry. They were announcing here Bayer products in the US, aspirin among them, but quite interestingly, there's another product, heroin. Heroin was developed by the very same chemist as aspirin, Felix Hoffmann, only 2 weeks after aspirin. After acetylating salicylic acid, he acetylated morphine, and you all know that heroin became a very successful product, but not in the pharmacies. So there's good reason to be careful what industry wants to bring to the market. There is need for regulation setting certain limits. And with these type of events, this is another one which prompted the increase in testing demands in toxicology. This was Contagan thalidomide, a substance which led to about 7,000 children with misformations of the limbs. I was born in 1963. This was shortly after the peak of this scandal. I had 2 of these children in my class. So this was a very impressive encounter with problems of medicines, of consumer products, which pose certain health risks. So and this is one of the reasons why, since the mid-60s, we have quite a focus on reproductive toxicity testing in order to have never again something happen like this. But there's also a sheer number problem. We have far too many chemicals. Chemists are so inventive. They have synthesized about 70 million chemicals so far. That's the number of chemical abstracts. 56 million substances have been registered with a CAS number, so they are inventorized. We have at least 100,000 synthetic chemicals in consumer products. And only a very few of these, about 2,000 to 3,000, have been extensively tested. So for the health endpoints which are really of interest to the general population, like cancer or reproductive toxicity, many more have been tested on acute toxicities or topical toxicities. But our machinery is at the moment maximum capable with the current testing paradigms to handle about 300 substances per year. No way to get to the 100,000 backlog, no way to address mixtures of substances, no way to address natural substances. One plant extract can contain 40,000 substances. There's no way to address these with the current testing paradigms. So how do we regulate so far? So Regulation 1.0, the Toxic Substance Control Act from 1976. It has not had any major amendments since. It did ask for screening of existing chemicals, but hardly anything of this has taken place. It puts all the burden of proof with the Environmental Protection Agency. So 55,000 chemicals were registered with the implementation of TSCA. These are the old chemicals, and you can say that almost nothing of these chemicals has been really properly tested. Since then, pre-marketing notifications are necessary whenever a substance is going to the market, and then EPA has 90 days time to ask for testing demands. And quite amazing, only 15% of these pre-marketing notifications have any toxicological data. Any. Hardly anything has broad toxicological data. And only in 200 cases of the 24,000 submissions, EPA requested additional testing. So we don't know really a lot about most of the substances which are on the market. And to quote a colleague of ours, Lynn Goldman, at the time EPA Assistant Administrator, she said about TSCA, our available tools for gathering testing data about these chemicals are cumbersome. It's almost as if we have to first prove that chemicals are risky before we can have the testing done to show whether or not the chemicals are risky. So it's a tremendous problem to get information in this regulatory framework. So let's have a little bit a look into the 2 areas which are of main concern, reproductive toxicity and cancer, as examples. We don't know how well any of these animal species which are used do predict actually the effect on pregnant women, on the embryo. on the development of the newborn child. But quite interestingly, if we compare these different species, if we run the same toxicity tests on mice, rats, hamster, guinea pig, the correlation is only 50 to 60%. So mice and rats do not predict each other better than this. And there's no reason to assume that any of these species would predict humans better than they do. For cancer, we have only data comparing mice and rats at hand, and we see that the correlation is just 57%. So a mouse does predict cancer, carcinogenic properties of substances in the rat only in 50% of the cases, showing the limitations of species extrapolations. And we do this with animal tests. The cancer bioassay, for example, is a test which does last 18 to 24 months. Can you imagine the delay until you can take a decision on a substance if you want to test? Because it's not only the treatment times, you have also to analyze, to prepare, so it's easily a 4-year study. It does cost for one substance more than $1 million in one species tested. It uses 600 animals, and as a result, we get 53% of all the substances which have ever been tested positive. Estimated in humans is 5 to 20% of chemicals maximal are carcinogenic. So we have a lot of false positive results as the result of these expenses. Reproductive toxicity is not better. It is shorter and cheaper, only about $600,000 per chemical, but it uses far more animals, and the positive predictive rate, as we were able to show both by theoretical assumptions and by going into databases, is about 2/3 of the substances. While the real positive rate is estimated by experts to be in the range of 2 to 3% only. So again, a tremendous number of substances which are falsely labeled as reproductive toxicants in order not to miss these 2 to 3%, showing the limitations of current tools. So to go a little bit more in detail quickly, just the cancer bioassay results. From all the chemicals tested in rats and mice, 59% were positive. And this didn't matter whether these were synthetic chemicals or naturally occurring chemicals, 57 or 60%. Even among the drugs which have been put on the market without cancer testing, about 50% of them were found positive when tested in 2 independent studies. Even in our coffee, you can see 21 out of 30 sub-ingredients of coffee, 70% of them were positive. So are we every morning brewing a carcinogenic cocktail to improve our health? If you take a look, actually, despite all these carcinogens which are without doubt contained, coffee consumption is actually reducing the incidence of liver cancer, at least in this study. So you can continue to take your favorite drugs. There is no indication that this is contributing to a major health effect. And this demonstrates a little bit the backside of the precautionary way we are testing at the moment. So let's talk a little bit why regulatory toxicology version 1.0 leads to these problems. Over-labeling, far too many false positives. The first reason is we are testing at high doses, while humans are exposed at low doses. These are human exposure data of a couple of common substances which were regulated because of the toxicities. And these are the animal testing doses. So most of them are several log orders below what we are actually doing in the animal experiment. And just to show the limitations of extrapolation, this cartoon I liked a lot. This is a newlywed couple, and as extrapolating from yesterday to today, he says, as you can see, by late next month you will have over 4 dozen husbands, better get a bulk rate on wedding cake. It is extremely difficult to extrapolate from high to low doses. It is producing, obviously, errors. The other point is, I think, very well explained in this clinical study. And this is the problem of multiple statistical testing. It is a study which was done by colleagues from Canada who did take the 10 million inhabitants of the region of Ontario. They took, in the year 2000, all between 18 and 100 years old. they randomly assigned them in 2 groups of 5 million, and they sorted them by the 12 zodiac symbols. And then they tested for 223 of the most common diagnoses for hospital admission. We're asking, is there any zodiac symbol which is leading to hospital admissions? And they found in total 24 significant associations, and they controlled for multiple statistical testing. And then they took the other 5 million inhabitants, and they tested these 24 hypotheses, and they still found that Leo is prone to have gastrointestinal bleedings, and that Sagittarians are highly significantly showing humerus fractures as reason for hospitalization. And their conclusion was very clear. They said testing of multiple non-pre-specified hypotheses increases the likelihood of detecting implausible associations. So if you only test enough, you will get some crap results. And what does this have to do with toxicology? The problem here was that they tested 223 different questions in one cohort. And despite all the statistic precautions, they did result in obviously something bizarre at least. But what do we do in toxicology? A regular 28-day study has 40 endpoints, so asking not 223 but 40 questions. A cancer bioassay has 60, and a 2-generation study for reproductive toxicity has 80 endpoints. So we should be, I think, not surprised that we end up in so many false positives. And for somebody coming from pharmacology originally, it is actually, for me, has always been surprising that in pharmacology, in order to find one positive effect, a very clearly specified principal endpoint of a study, we use thousands of subjects and good statistics. And we only ask one question. And we come to a result, hopefully. But in toxicology, to get the negative effect of one substance, which is nothing really different, we rely on groups of 20 to 50 animals per group. We test dozens of endpoints, and we use lousy statistics. This is something which does not go together well. And we can see that we don't have the area under control. Computers or experts you can test, they were asked to evaluate substances before they were tested in the cancer bioassay. These are individuals, in the small letters, in the capital letters. These are programs which have been asked to predict what is going to happen. And you see this is the 50% true positive rate, and this is the false positive rate, 50%. They're all grouped somewhere here. So that's more or less the outcome of expert knowledge which we have accumulated over 40 years. And even with these precautionary measures, as our colleague Alan Silvergate pointed out rightly, we cannot get all of the important carcinogens. There's still no animal model for cigarette smoke-induced lung cancer. There's no rodent leukemia by benzene, or there's no genetic mutations in animals by arsenic, just to take very prominent examples, despite 10 times more substances falsely accused in these testings. So we have a couple of problems here. We're using maximum tolerated doses. 10% of the animals die from the treatment. So this means necrosis. Necrosis is inflammation. Inflammation is proliferation. And proliferation is one of the strongest promoters of carcinogenic effects. We have the multiple testing aspect. We have the cost and the throughput problem. We have a problem that only 57% concordance exists between mice and rats. And a very interesting study did something very simple. They modeled if we would increase the number per group from 50 to 200 animals, we would result not in 53% of the substance being carcinogenic, but in 92%. This shows how we accumulate false positive results in our study design. Or you could say a chemical which is not positive in the cancer bioassay has not been tested long enough. So at the same time, we're trying to regulate on this basis. And a very good example is dioxin. Dioxin has been regulated on the basis of cancer studies. And we have a reference dose, an acceptable dose limit. Bruce Ames, already 20 years ago, did say, if we do this very same calculation on alcohol, we could drink one beer in 345 years. This is about the calculation we are applying, the threshold calculation for dioxins. And this shows some of the problems of toxicology version 1.0. At the same time, it has created an enormous chemophobia. Here on a cosmetic product warning, this product contains 16 toxic and 8 carcinogenic ingredients, while experts say it's maximum 2% of all cancer cases which are a result of chemical treatments. So we have also frightened people with these results. Because this gentleman here, Paracelsus, he got it right. The dose makes the poison, but we don't get this message passed. We can have these various substances to be exposed. At the same time, let's say I jump a little bit. In the end, there's the wish to act to reduce environmental cancer risk. At the same time, we are somewhere at a dead end with the type of methodologies we are using. We need something new, a version 2.0 for toxicology. So we're a little bit in the situation of this guy who is looking under the street light in the night for his keys. After a while, somebody comes and says, "But did you lose them here?" And he says, "No, but here I have light." We're using tools for which we know that they are not adequate to find the keys we are looking for, but we use them because we have them. They're the streetlights which are available, the cancer bioassays, the 2-generation studies. We know their limitations. We don't have to dig deep in the literature to find them, but we use them because we have nothing better. So we're using technologies which are outdated. And this is the development of the telephone. And this shows you on this same time scale when our technologies for safety have been developed. In the 20s, the LD50 test for acute toxicity, chronic toxicity testing. In the 40s, eye and skin. The 60s, reproductive toxicity testing. Around 1970, the cancer bioassay. Every technology has its time. Or every time has its technologies. Also, our modern technologies will phase out somewhere, but not in toxicology. And if it's true that the knowledge in the life sciences doubles every 7 years, then we have more than 1,000 times more knowledge now than at the time when we designed these tests. We have to bring it only into action. And this is, I think, where it started, or where, let's say, a lot of these various reasons of uneasiness with the current testing paradigm came together. This report of the National Academy of Sciences from 2007, and Jim is here, Jim Yeager, who was part of this committee. I think this report has really created an atmosphere of departure, now to move towards a new type of regulatory type of testing. It certainly should incorporate the lessons learned on the way by trying to fix some patches to this system with alternative methods. But the most important suggestion, I think, is to move to the new technologies from biotech, from the bioinformatics revolution. And the principal proposal here is to base this on mapping of pathways of toxicity, to move to mechanism, to move to understanding why toxicology, how toxic effects do occur. And to illustrate the excitement, only actually half a year later, with the first author, Francis Collins, an agency collaboration was announced in Science between EPA, the National Toxicology Program, and the Center for Genomics Testing. And they said, following the lines of the proposal, we propose a shift from primarily in vivo animal studies to in vitro assays, in vivo assays with lower organisms, and computational modeling for toxicity assessments. And Francis Collins cited by USA Today, toxicity testing was expensive, time-consuming, used animals in large numbers, and didn't always work. A very nice summary of what I tried to do to tell you in the first half of the presentation. And Elias Zerhouni, his predecessor, said animal testing won't disappear overnight, but the agency's work signals the beginning of an end. So there is already something moving in the regulating agencies. And in fact, in 2009, EPA made this their new toxicity testing paradigm. So something happening here. But how? And Paul Simon had this wonderful song, 50 Ways to Leave Your Lover. I think the important message is there's not only 50 ways to leave your lover, there's only a couple of 100 ways to kill a cell. And this basic assumption, I think, is the basis for doing something new. We have been working in very many areas of toxicology, identifying a pathway here, a pathway there. But the idea that this is a finite number, I think, is making the difference, because we could come to an end at some point. So the concept of pathways of toxicity, the idea that a given toxin in a given cell type engages certain pathways to come to a certain phenotype, a hazard. And another toxin might employ some of them, but is not leading to the same phenotype. And there will be overlaps between cell types, and there will be overlaps between different toxins. But if this is a limited list, we can start, first of all, to understand what is really required. And then we can start to map this and annotate this. So we can identify what pathways of toxicity are necessary that a certain hazard takes place. What are toxin classes which are involving which pathways? Which cell type does represent which pathways of toxicity? What is the reason why the liver cell is sensitive and the cardiomyocyte is not to a given toxin? Or what are species differences? Which of the pathways is reflected in rabbits, which one is reflected in rats, and which one is actually found in humans? And most importantly, we should understand that actually most of the chemicals are not really toxic. If you look into the new industrial chemicals which were produced after 1980, 90% of them were not acutely toxic. We could not kill rats with up to 1 gram per kilogram of these substances. 97% were non-skin corrosive, 93% non-skin irritants, 97% are not teratogenic in humans, 80 to 95% not carcinogenic, and so on. So most of the chemicals are not really having a hazard. So the idea is that we need actually to identify concentrations of substances or substances at all which do not trigger pathways. But this information gets only relevant if we have a comprehensive list. Because you could always say, okay, it does not trigger a pathway of toxicity here, but it could do another cell, it could do it under different testing conditions. Only if you have tested at least the most important ones, you can be reasonably be sure that this is no longer representing a problem. And to give you a little bit of a feeling what this is, there is an area, developmental toxicology, which is actually developing very nicely, though we have this big problem with reproductive toxicology as a whole. And the National Academy of Science report from 2000 did suggest, for example, that there's only 5 major pathways which, when disturbed, lead to problems in early development. So we took this up, and in a collaboration between my group in Italy and Professor Michael Schwarz in Tübingen, we just took one of these pathways, the Wind pathway, and we produced a reporter gene, a cell line, which was indicating of a disruption, any disruption of this process. And we put this transfected mouse embryonic stem cells. And this very simple test allowed us already, with one pathway only, to identify several of the important human reproductive toxicants like lithium chloride, retinoic acid. We could show the different potency of various valproic acid derivatives, and adding a metabolizing system, also the teratogenic effect of cyclophosphamide. And this is one of the hundreds of pathways only. But this is the type of simple test which could be designed and which could be automated if we once come to such a complete list. Here in the school, our own work is focusing on developmental neurotoxicity, a very interesting area for regulation. People are concerned about autism, about hyperactivity. Animal testing is the solution, as always. In 2007, only OECD guidelines were established, but it does cost 1.4 million to test one substance and 1,400 animals. So what the people in our lab, Helena, Erwin, Sabrina, Tekum, and Shelley, are doing at the moment, they're trying to use mainly mass spec metabolomics and integrating genomics data in order to identify pathways of toxicity doing this here as a pilot application, so that we have new pathways which we then can translate in other systems. And this nice machine just arrived at the beginning of the year and is now starting to work to allow us to do the metabolomics. And what we are doing is, in the first cell system, we call them mini brains. These are a couple of thousand cells which form aggregates. And we treat them with known developmental neurotoxicants like lead or trichloroethylene. And then we see, first of all, that certain metabolites change. And with principal component analysis, we can see how the different concentrations of the substance do cluster, give typical response patterns. And we can start to compare, we can start to identify the pathways behind it. That's forward-looking. And to show you how regulation could be done on this, I borrowed a couple of slides from Bob Kavlock from the EPA Toxcast group, which is, I would say, it's fair to say, spearheading some of the developments in the agencies. They are running a large program, Toxcast, which is using pathway-based assays and high-throughput testing. They're checking hundreds of substances in hundreds of different tests, but they're using from-the-shelf available high-throughput tests. I cannot go into details. These are some of the endpoints they're looking for, and they use their bioinformatics and machine learning to understand and interpret this. And to give you a case example, they had a problem last year. Let's say everybody had a problem last year because of the oil exploration, the platform in the Gulf, which did spill oil into the Gulf. They had the problem that BP wanted to use emulgators, and there was no experience with the toxicity of these emulgators. And they actually did use, in the end, 1.8 million gallons of dispersant on the surface and subsea. And it was necessary to make a decision fast what type of substance could be used for this purpose. So the EPA was concerned about this, and they asked, can we do something with these new tools, high-throughput type of testing? So they put this into their 460 assays, and they characterized the different dispersants which were discussed with the various assays. And I will not go into any details how they took their decisions, but in the end, it was possible within 4 weeks only to make a complete evaluation at cost of $12,000 per substance, which would have taken more than a year and cost about $500,000 if you would like to take the same decisions with traditional approaches. So this was ad hoc, this was pragmatic, it was certainly not based on the latest scientific state-of-the-art knowledge, but it shows how in the future this type of information gathering could help take informed decisions where we are not able to provide better data in the same time, and certainly not at these costs. So the vision is that we can result in a new type of toxicology, that omics technologies, image analysis, robotized and automated testing, the new technologies made available over the course of the last 10, 20 years, give now new situations of high content and high throughput type of testing. So either a lot of information on one substance or a tiny piece of information, but on many substances in a short period of time. That this creates an information-rich situation, not the number of limbs of the animals counting to the sky at the end of the experiment divided by 4 as my result, but an information-rich situation. That we can use bioinformatics and data mining and the knowledge on pathways from biochemistry, from molecular biology, from mechanistic toxicity to develop a systems toxicology approach, which is allowing us to map the pathways of toxicity, the comprehensive list of pathways. So this is calling for a type of human toxicology project. And this is obviously something which is borrowing from the Human Genome Project. And you have seen most probably these people, Francis Collins and Craig Venter. And quite interestingly, I think it is fair to say that the project really took off only when this guy, who's for some good reason, I think, in front, was coming into the play. It was very strongly the private competition which did drive the whole exercise. But it was necessary that Celera became the company which was created to run this, became accepted by feeding into public database, not making it an exercise which was looking for profit only. So at this moment, there are strong consortia trying to develop something like Hugo for toxicology. A human toxicology project consortium has been formed, but they are calling for public money. So we're starting to think whether we could possibly set up something which has, let's say, making use of the corporate resources to get something done. And at this moment, we are partnering very strongly with Agilent Technologies and trying to work together, think together what we could do to set up such type of consortium. We want to engage big pharma, chemical industry, and others to join partners from academia with their knowledge on pathways and these things to start a collaboration. And the idea is that new technologies and bioinformatics are brought in, that measurements need to be executed, obviously, but we also need conceptual work. We need the concepts of pathways of toxicity. We want the failed substances from pharma development and their predictive models, but we also want their resources. And, as I said, biological samples and pathway expertise from collaborating partners. So the vision at the moment is to create Potomac, the Pathways of Toxicity Mapping Center. So a project which is actually setting this into action, starting with the very first pathway in the very first cell system with the very first toxicant. And the idea is to start with those systems in which a lot of investment has gone, which are the validated alternative methods. We have about 40 of them after 20 years of work. This has been about $300 million of investment into development and validation. But for these validated alternative methods, we have robust protocols. We know that these are good cell models. They already have achieved regulatory acceptance. We have reference substances. We know for which or which not they are predictive. And we know where the threshold of adversity starts. We know we have cut-off levels to say this is a pathway, this is an adverse effect, and this is not. And there's a couple of these systems which we are at the moment prioritizing. We don't need to go into details, hoping that they will tell us the first pathway and that we can learn by using them. There's challenges around this. Nobody has an idea how many pathways there really are. I think that most of us are still thinking far too linear in our pathway thinking. It is a network which is disturbed. It is not one event follows the other. We have far too much linear thinking here. We want to start by combining two technologies, which is metabolomics and genomics. But we will soon need others like proteomics, phosphoproteomics. We have not solved the problem how to annotate a pathway. How do we describe the elements necessary? We have no idea how we validate our results, really. And also, if we want to share, we don't know how to govern the public database which we want to create. So on the one hand, we have a blue sky project, some start into the 21st century. We hope to achieve some technological leadership by doing this, to accelerate the paradigm shift, which is so urgent. And we believe that some substantial progress can be done already with quite a limited effort in the very first systems, using only a few technologies. And that this approach also is scalable because we can put in one system after the other using the increasingly developed mapping technologies. And we are discussing at the moment with EPA and the National Toxicology Program that they would host the public database and would make this then available. But there's challenges ahead. As I said, there's no concept yet. We don't know really how to define adversity. We will need many more components to make this actually work. A list of pathway is not yet a new regulatory scheme. And we need also a transition from the current system. And here, evidence-based toxicology comes into play. Some of you will know that I'm heading here the first chair for evidence-based toxicology, which has the idea of bringing principles of evidence-based medicine into toxicology. I have not the time today to talk about these concepts to major extent, but it's part of the paradigm shift, and 5 days since the last paradigm shift. Evidence-based toxicology has been proposed by us in 2006, and already in 2007 we have had the first international forum for evidence-based toxicology, which was starting to develop a collaboration towards trying to employ evidence-based medicine tools in toxicology to evaluate the existing and the new tools in toxicology. And I think quite importantly, next week we are kicking off the evidence-based toxicology collaboration on occasion of the 50th Society of Toxicology meeting in Washington. And we have gathered a steering group here from various agencies, industry, and opinion leaders who will, with us, try to get started a process which helps to open the doors and help to endorse new technologies with evidence-based medicine tools to get the new toxicology running. And with the help of an anonymous donor, we have raised the funds also to run the secretariat for this collaboration here at Hopkins. So I would like to close first with this cartoon, which I like a lot, because also as there's limitations to the new tools, there's also limitations to the concepts of evidence-based medicine. What we must avoid is something like this. We have to catch him, but that's not evidence-based, which is the backside or the downside of a lot of the evidence-based medicine when it is abused to not treat. We do not want to create a situation where evidence-based toxicology is used to not regulate substances. We have to continue to use the existing tools until we can offer something different. So evidence-based toxicology is not meant to attack the existing without at the same time being a sparring partner for the development of the new tools which are necessary. So closing with my favorite quote from this guy, Victor Hugo, who said, "Nothing is more powerful than an idea whose time has come." I strongly believe that the time for regulatory toxicology version 2.0 has come. Thanks.
So Victor doesn't look very happy in that sector. I think so let me ask the first question. So this idea of mapping all the pathways of toxicity and then substituting that for animal testing. I have to say, not knowing the field well, it's just hard to believe that the way you describe the methodology that it's a regulatory method. I believe you, but it's just not good. But wouldn't you have to know all the pathways? I mean, because you couldn't substitute it unless you were fairly sure you had. If you think the animal model is the analog, right? You just dump it in there and you let the animal metabolize and see what happens. You're going to a digital model where you're isolating each pathway. You have to know all the pathways to do this work. So.
Yes and no. I don't think that we need
all the pathways, because there will always be, we can only approximate to get to a list which is comprehensive. And the question we can only pose is, do we find the bad guys of the past? If there is tomorrow a substance which makes you lose your fingernails, nobody has ever thought about this as a toxic effect, and nobody will have thought about the pathways of toxicity of this, and we will be blind spotted for this type of toxicity. So nobody would think of endocrine disruption or atherosclerosis being a toxicity endpoint until nanoparticles produced an association with atherosclerosis. Yeah? We only can look for the known ones. But the current system does not show us very many of the problems we know. Thalidomide would still not be caught by the standard test in the rats because it is not a reproductive toxicant. But we can be sure that we can test in such an approach to an extent that we get the problems of the past and we are prepared for those new ones.
Okay. So comments or questions? So Jim, I'd like to ask you, so is it really, it's all due respect, is it really that bad? Or is it really such a, I mean, regulatory framework so stupid? I mean,
Yes, I think so. It's just been adding, as Thomas tried to indicate, adding on test on top of test to try and determine if a compound is going to pose a potential risk. The problem is that the predictability of the animal systems for humans is typically unknown. You know, with carcinogens, when they tested all known human carcinogens in animals, these are the ones that caused, you know, bladder cancer in humans and liver cancer. Well, of course they were carcinogenic, but the opposite we don't know. How many of these compounds that are being detected in the animals are really human carcinogens, especially when the doses that are being given to the animals are logs higher than human exposure of OB. So the extrapolation line, we have no idea how to do that extrapolation.
but the issue of multiple outcomes, I mean, you just need a statistician there. I mean, right?
And you can either start increasing the groups of animals to a couple of hundreds per dose group, which is economically and practically not feasible. Then you could satisfy statistics, or you need a different approach. Yeah? In vitro repetitions are not an issue. Yeah? You can sort out the noise from the real ones.
Barry, and then Tom.
So if rats and mice are different, how do you choose cell types? They're wildly different from one another. How do you know how to do that? And then how do you know from a given cell how that's actually going to work when you stick it into a body, whether a human or a mouse or a rat or anything else? How do you do that?
I mean, this is very much the concept of classical alternative methods. Find a good cell model and replace the animal with the cell model.
What's the definition of a good cell model?
It is one which is shown in validation to be predictive for a certain part of the chemical universe. So it gives a good correlation. The new concept is completely different, because even a lousy cell model, which reflects only one pathway of toxicity, which is not sufficient, can be a very precise and important information if combined with another lousy cell model, another reporter gene or whatever, and if we only know that these two pathways are critical, and we need to be careful of substances who are capable of triggering either or both of them at the same time. So to be even more reductionist, but being comprehensive with the mechanistic foundation, I think offers us the opportunity to get away from phenotypic or apical type of animal test to apical type of cell models, where you always raise exactly your point, what would be, how to choose the right one. Yeah?
So Tom,
So there are some in the field who make exactly the opposite argument, that by going toward the pathways, we're losing the goal, which is public health, which is population health. And so there might be epidemiologists who are saying you're forgetting about the big picture of cumulative risk and how it affects the human population, or how there's tremendous variation and susceptibility in the human population. So if you look at the epigenome and epigenetics and the individual variation there, I'm not quite sure how this comes into play. And in fact, just the other day, I heard an eminent toxicologist say, this is not ready for prime time and may not be for a long time, and yet we have to make decisions now. And she urged that perhaps we shouldn't be so worried about the pathway that an organ or a system gets disrupted, like the endocrine system, but the fact is that if there are multiple insults, it doesn't matter if the hormone is changed by what pathway, but the fact that it is. And I was just wondering how you counter that, because as we pondered this approach at the National Academy, thinking about risk assessment, Mal Anderson, who was on one of your slides, said it's just not ready to really guide decision making yet. So I was wondering how you might comment on that. So it seems like the field's going in two different directions for the future.
I mean, in an ideal world, we would do tests on humans, and we would use mainly epidemiological data to find out about risks of substances, because that's the most relevant information. But it took us 50 years to show that smoking produces lung cancer following these approaches. They're better, they're getting better too, and I don't argue against maintaining. And I think we need to continue catching the falling guy. We must not use, say, where we have no evidence, we don't act. Because it's, we enjoy relative safety. It is only that we cannot really move the current system by evolution. I believe that we have a tremendous opportunity at the same time on constructing something new. Because it will be certainly difficult to take the possible noise of some type of biological effects of a pathway being triggered to take a regulatory decision. But we can only understand after we have learned which are the crucial pathways and which are those which are, if triggered, really suspicious, that we follow up on something. But if it is true that most of the substances are not toxic, or we're looking for the concentrations, to establish concentrations where they are not triggering anything, then this type of approach is extremely helpful, as long as we can test enough of these, and we are confident that we have a comprehensive list. So I think the beauty of the approach is really that it is bringing into a field which has been uneasy for many years and is trying to optimize the current tools, the streetlights which we have, to make them brighter, to see a little bit further, to find the keys, that it is bringing in a completely new component which is saying, yeah, if we had the human toxome mapped, we could start constructing batteries which can test so fast we get the information, and we will be able to design, engineer a different system. Then we might compare results, and we will see whether this is better, cheaper, more predictive.
This goes back to Dr. Zirkin's question, and it relates to Thomas's slide about the lamppost. What I've seen is that many times people pick cell lines because they are available. They don't compare them to a human cell line to see if they are, in fact, a very good model. So they just pull it, they say there's a cell line, we take it, we use it, it may not be right. Then when you start looking at some of these pathways, people start measuring what they can measure. And that's where a lot of this has come wrong. So we have, as Thomas has been trying to say, we have to be a little bit more intelligent and think about how we approach this to make this actually work. Choose your cell lines carefully, look at your pathways. Just don't measure what you can measure, but think about what we know about biology as it's now currently available. Because that's the problem that we've seen. Too many things are measured and have read it up with blind alleys. So we need to think about how we can use the new technologies, understand them, and apply them, and have used in vitro systems. And when you get the in vitro system, then you could go and test that in an in vivo model and then find out if it's actually true. Because you can actually do some comparisons between rodent cell lines and human cell lines to say, is the rat cell like a human? If it is, you use the rat model. If the mouse is that like the human, use a mouse model. So we just have to be a little bit smarter.
Yeah, just a follow-up.
Please, no, go ahead.
Just to follow up on Tom's comment, and it's not ready for regulatory toxicology at this point. But I mean, Thomas is, and it's gone through a lot of this very fast, and there's so much more behind it. And even if you look at the cell lines that were listed out there, they are carefully selected. Number one. Number two is that part of all of this is the pharmacokinetic, if you will, modeling, so that you can go with these concentrations that you're exposing these cells to, which are really going to be able to be low concentrations, and then model that. That's an area of Mel Anderson's expertise and others. Model that to what you would predict the human exposure would be at the tissue level, knowing the all of those PBPK modeling approaches, which are going to improve dramatically, I think. And one key thing that we all have problems with is the metabolism. How do you expose cells to metabolites, and what are the best systems? But these are all things that have to be really refined as this moves forward.
Which one is the best metabolite? Because some things
Correct.
have several metabolites that are potentially toxic.
Correct.
I mean, I carefully titled this presentation a sneak preview of the beta release. So it is meant really of, let's say, raising interest in something which is going to be tested. Yeah, it is not something which is going to be released as something which is a definitive answer to replace version 1.5, which we are enjoying at the moment. It is, but I think the key take-home message is really there is a potential of coming with a new quantum leap type of approach by coming to mapping the human toxome. This is completely different. It is putting in essence on speed the whole pathway identification, the mechanistic toxicology, which has been almost neglected by regulatory toxicology.
I had a question about, I'm curious particularly about pathways toxicity, and it's obvious that opportunities abound there. Do you think there are a finite number or finite classification of toxicological pathways that you think are generalizable enough to go across large categories of chemicals?
I mean, I would say it's a philosophical question here. I believe to some extent, let's say, first of all, I believe that the fact that we see signatures in these new technologies of certain classes of toxicants is highly indicative of common pathways underlying these signatures. Whether the number is really finite or reasonably small, that we can actually do a comprehensive list to construct later tests is, as I said, philosophical. I believe that Mother Nature has not left too many ways of killing us, because otherwise we would not turn almost 100 years now. That's the life expectancy of a newborn baby. 50% will live up to 100 years while introducing more than 100,000 chemicals to consumer products. So obviously we are a pretty robust species, yeah? Which might mean that there is not very many ways to kill us, which is my optimistic interpretation of this all.
Really a practical question of like, well, all right, which do you consider based on what data that they provide you, or not provide you, or from a regulatory perspective?
I mean, as I said, at the moment, I'm not suggesting that the map of pathways is already giving us a regulatory framework. Yeah? It's very important to distinguish these. But I think it will be a pillar for building one. Because only when we have this list, we can design a testing battery. And when we design the testing battery, we will have to solve all the problems which were mentioned in the discussion: metabolism, in vitro to in vivo extrapolation, human extrapolation, a lot of issues here. We will have vulnerable populations to solve. But I think that such a tool is like the human genome. The human genome did not help us to treat any disease by being available, but now its applications, all the new things we can do, help us to gives us new tools at hand. And I think that in a very similar way, the list of pathways would help us to accelerate progress in toxicology to change also the regulatory part.
So maybe last question.
I am very impressed with your vision for this, but I'm wondering, you mentioned about a current limitation is mixtures of chemicals, and we know that some chemicals affect the toxicokinetics of other chemicals, and I'm wondering, do your vision of a simulation down the line, how are you going to account for the uncertainty in the chemical mixture of how things are going to be metabolized, how they're going to be processed? And because my understanding of simulations like the ones you're proposing is it's a deterministic rule, and if you aren't sure about how things are going to map, how are you going to do networks of chemicals?
I think this is a very important question. This was a very nice last question. First of all, I think that mixture toxicology cocktail effects are a key problem, and the current framework does not allow us to address them at all. And I don't believe that by moving to cells we really have a chance to do this better, because we are amplifying also the problems, the limitations of the reductionistic systems. And you can test an endless matrix of not only concentrations of two substances or more, but also of timing of the events, when is which substance given, and so on. And this gets endless and is almost impossible to interpret. But I think if we would understand the pathways, we would understand that a substance A and a substance B are triggering the same pathway of toxicity, and then they will typically be only additive, and we can evaluate this. If we have an understanding which second pathway is amplifying the first one, then we see that the two substances have a possibility to synergize, because A is triggering the toxicity pathway and B is triggering the amplifying pathways. But this is part of the annotation process, which can only start if we have really a reasonable list of these pathways at hand. And I think the big hope is really that we are, in the end, bringing the academic advances over the last 20 years finally also into the regulatory arena by making them tangible, by making them applicable, by having such a point of reference as a human toxome map.
Great. Well, let me ask the last question. Are there any Leos in the audience? Because we're going to have a reception, and I don't want you drinking wine, because I don't want any GI bleeding out there in the reception also. So, Thomas, that was a great talk, very thought-provoking as always. Thank you.